Today we are covering chemical synthesis, chemical amplification, and sequencing. So I just mentioned earlier that DNA is a chemical molecule. It's an organic molecule. It has carbon, hydrogen, oxygen, nitrogen, and phosphorus. Right? And so this is a chemical molecule with biological functions, and these functions are very important because transcription is important for protein production. Proteins are the major workforce in our cells, and there are almost last time I read was at a time there could be typically 22,000 to 25,000 different proteins in one cell. And replication is important. Replication is directly coupled to cell division or cell cycle, because replication takes place in the S phase of cell cycle, and that leads to the duplication of DNA chromosome, and that could be important for mitosis as well as meiosis. And if you go the meiosis way, in anaphase 1 there is the homologous chromosomes depart into different daughter cells, the reduction, and that's what goes on to the next generation. So the fingerprint of one generation is passed on through DNA, right? And majority of the DNA is similar. For example, my hemoglobin gene could be very similar to all of yours. But there could be some patches, some islands of DNA that could be very different from me and you. And there used to, in normal times when we used to meet in the classroom or lab, we used to do an experiment where we used to look at some of the genes, for example, ALU gene, and whether this is present as homozygous, or it is present as heterozygous. Right? They're present as homozygous or heterozygous. So this can be used for looking at epidemiology. For example, we can look at in one zip code or one in one of the boroughs of New York City, that how many people are there with homozygous ALU gene or heterozygous ALU gene. Okay, so as I mentioned to you, coming back to it is a chemical molecule. Now, giving it as a chemical molecule, and if we have the detailed knowledge of the chemistry of DNA, right? Chemistry of the DNA, then can we synthesize this molecule in the lab, right? And if we synthesize this molecule in the lab, then it could be helpful to do research, understand the function of DNA, and it can also be used to develop tools which could be used for many, for example, many purposes like gene therapy, vaccine, so and so forth. And so that is the main goal of this chapter, to introduce to you or to teach you how DNA can be synthesized in the lab settings. And this newly synthesized DNA molecule can be used for many purposes, right? And when we finish the synthesis, we'll also talk about synthesis of mRNA that is being used for the purpose of developing COVID vaccine platform. Now, one thing interesting, I was watching a video last couple of days, and it says that RNA is one of the most sensitive molecules in our body. Sensitive means it is very susceptible to degradation. It can be destroyed very easily as compared to DNA. DNA, you can leave it on a bench and forget it for several years. You can come back and start working, doing research. It could be part of fossil, million years back or 1,000 years back, but RNA is very, very delicate. So what these companies have done, that they have modified mRNA to make it more stable. But when we finish this chapter, then in the end we will talk about mRNA, because then it will build your base to go there. So the objective is that learn about how DNA is synthesized, and then one of the tools of modern biotechnology is polymerase chain reaction, and the third is DNA sequencing, right? DNA sequencing is at the heart of finding out the variants, right? The variants of COVID-19. Without sequencing, we cannot find in which way the pandemic is going. So, any one of you who believes in starting his or her own business, these 3-4 technology, unless you know the theory, you can't, do a practical or practice. So if you understand the theory, then it makes you comfortable to do the experiment. And any one of you who has, a desire to open a company, these 4 techniques are good enough to start a small company in a basement, or in a garage, or in a closet. Nothing that you need more than this. And most of the biotech companies, which started 10 years back or 15 years back, they started with these 3-4 technology. And now when the COVID came, when Zika came, when other SARS virus came, and people needed to sequence these genomic, now we have information from human genomes. So sequencing has become a part of diagnosing cancer also. So these companies that started as a very small company in upstate New York or in New Jersey, they have become very big name now. One of those companies are GeneLink and GeneWiz, and so on and so forth. So, these are very important techniques. So let's, I always, I emphasize during while we're talking about the syllabus, that I'll go and touch base the basics of molecular biology before diving into the topics of this course. So as the story is about DNA, so let's look about, I might be repeating this, but just in case, if you have missed it, then you can, or even if it is a revision, it doesn't hurt. So, so DNA, it's a very architecturally a very perfect molecule, although it looks, very, very squiggly or wavy, but it is very tightly packed. Almost meters of DNA is tightly packed inside our nucleus. And this is also tightly packed with the help of proteins. And the alphabets, there are only 4 alphabets in the DNA. In English literature, you have 26 alphabets, A to Z, but here you have only 4. And these 4 letters are, as compared to English, these 4 alphabets, these 4 nitrogenous bases in DNA is enough to generate variation among tons of species that is present on this planet, right? These 4 bases. So these 4 bases, and one of the most essence of DNA is its stability, right? So the stability of DNA comes from 2 things. One is that the bases, the nitrogenous bases, they are arranged one above another. And rather, I would not say they're arranged, they're stacked one above another. So if you take, let's say, quarter coin, and you place 100 quarters, coin one above other, so all will be looking same size, same shape. Yes, in the DNA, there is variation between purine and pyrimidine, but they're stacked like coin. And that generates hydrophobic energy. And that is important for stabilizing the DNA. The second force is hydrogen bond. Hydrogen bond, if there was no hydrogen bond between two strands, let's say there was covalent bond, then the replication was just impossible. Because breaking a covalent bond, it takes huge amount of energy. Hydrogen bonds are weak bonds, and they can be broken easily. And proteins in the replication cycle, like helicase, they can easily unwind the DNA. They can easily free the two strands of DNA from hydrogen bond. So, the nature. So you can imagine that in DNA, which is the most important genetic molecule, one of the most important molecules because it does so much important activities, has hydrogen bond, and which is one of the weakest bond in chemistry. And this DNA has hydrogen bond. This is to facilitate replication, transcription, so on and so forth, because it is easy to break. Now, why it is easy to break, or what is the consequence of breaking? So breaking is essential because if the DNA doesn't break and it stays always as double-stranded, then it will be difficult to perform replication. It will be difficult to form, generation of mRNA or any other RNA, because these strands have to be linearized. If they're not linearized, then replication and transcription, because one of the strands of the two in the two strand, they have to work as a template for generation of daughter strands or mRNA or tRNA. So it was hydrogen bond, if it was covalent bond, then it would have been very, very difficult. And also remember this chemistry, that the second carbon atom in the ribose of DNA, deoxyribose, there is no OH group. There is no hydroxyl group here, right? Whereas in RNA, both will be OH. So let's move forward. And here you see that purines, adenine and guanine, they are bigger structure as compared to cytosine. And if you see the difference between uracil and thymine, they're very similar. Thymine is in DNA, uracil is in RNA. What is the difference between thymine and uracil? A methyl group, CH3 group, is present in the benzene in the ring at the 5th position of the ring. Methyl group. Right? That's the difference between uracil. Uracil creates a world of RNA, and thymine creates a world of DNA. And also, this is very important, that without phosphate, base and sugar, they're called as nucleoside. And nitrogenous base is attached to glucose. The nitrogenous base is attached to ribose through a glycosidic bond through the first carbon atom. Then one phosphate, monophosphate, diphosphate nucleotide, and triphosphate nucleotide. So this is the building block for nucleotides, right? So remember that the building blocks for nucleotide, a nucleotide which are how many types? 4 types: adenine, guanine, cytosine, thymine. So the precursor for these are GTP, ATP, TTP, and CTP in case of DNA, the precursor. Now, another important part of the chemistry is that look at deoxyribose, or let's just start with ribose, right? Ribose has both of them, deoxyribose and ribose, they have 5 carbon atoms each. But the difference is ribose has OH at 2 prime, 2 second carbon atom, whereas deoxyribose has only hydrogen. Now I want to introduce a very important molecule here. This is called as dideoxyribose. So second carbon atom without oxygen is called as deoxyribose. Whereas now carbon number 2, carbon number 3, if both of them lack oxygen atoms, then they are called as dideoxy. So number 1 is ribose, where both the hydroxyl group, OH group, are intact. Deoxyribose for DNA, where one of the oxygen is missing from carbon number 2, and dideoxyribose, where both the oxygen atoms are missing from the carbon atoms, second and third from the ribose molecule. Now, why? So this may not be available in nature, right? In nature, only 2 types are available here, but this has been chemically synthesized. Now, why this has been chemically synthesized? Because I remember I told you, I mentioned to you in my first or second lecture that this 3' hydroxyl group of deoxyribose or ribose, they are sterically very friendly, very approachable for the synthesis of DNA or RNA. So if you replace this OH group with H, then you're stopping the replication or transcription. Why? Because hydrogen is not sufficient to form phosphodiester bond. The third carbon atom in deoxyribose and ribose, they have OH group. Both can facilitate the formation of phosphodiester bond. But if there is only H, then there is no formation of phosphodiester bond. So if I use this as a nitrogenous base, right? So let's say this is dideoxy CTP. So dideoxy CTP will be that wherever this is there in the form of dideoxy, the chain synthesis will stop. What do I mean here? So let's say this is a one of the DNA strand, right? And for doing a synthesis, one of the nitrogenous bases is, I'm talking about artificial situation, is dideoxy CTP. So let's say in this DNA, at this spot there is nucleotide G, guanine. So wherever there is guanine, if let's say DDCTP comes here, this is guanine, this is also guanine. Wherever this DDCTP goes and hydrogen bonds with guanine, the chain will stop. The chain will stop because the ribose here has a third carbon atom. Instead of hydroxyl, it has hydrogen. So the chain will stop, chain will terminate. So these dideoxynucleotide is used for chain termination, to stop the chain. And why the chain is terminated? This is one of the important steps for sequencing. So Frederick Sanger was the scientist who developed the method for both DNA sequencing and protein sequencing. So he's one of those scientists who got Nobel Prize twice. So he was chemist, and for doing chain termination, he used this dideoxy. So let's go back to our slides. Now here, this has 3 chapters, but this year and last year I mentioned briefly, but this year I will talk in a little bit detail about mRNA synthesis for vaccine development. But essentially, this chapter has 3 topics: DNA synthesis, DNA sequencing, and polymerase chain reaction, PCR. So, and I just want to just go with the simplest thing, and that is polymerase chain reaction first. Polymerase chain reaction. So, so students, sometimes when I try to show YouTube video, sometimes the Zoom doesn't sync with the YouTube video. So I suggest you definitely watch this YouTube video. It's very helpful. But what I'm going to do is simplify as much as possible the principles and the functioning of PCR. Right. So PCR. Then after I teach PCR, I will teach DNA sequencing. And once I finish DNA sequencing, then I'll go for DNA synthesis or gene synthesis, and then I go to development of mRNA as a vaccine precursor. So the scientist who was, it's a very interesting story, but I won't go into detail. But the scientist who was responsible for the discovery of PCR was Carl Mullis. And Carl Mullis, or Carrie Mullis, you can say, he was a biochemist, and he did his bachelor's and from Georgia Institute of Technology undergrad, and then PhD from California. So there was, he used to, it's a very interesting story. You can read his Wikipedia page, but he used to drive often, and what he used to do is that when you're driving at a particular location, generally what happens that there are some locations that we always, whether it's a grocery store or laundry, we keep going back and forth, back and forth. So he used to drive in a hilly area, and he used to drive back and forth. So from this doing back and forth, he thought that if he develops some reagent which can go in the DNA back and forth, then this can be used to amplify the DNA from 1 copy to million copies, right? So PCR, polymerase chain reaction, is a technique or technology that can be used to amplify DNA from 1 copy to millions of copies, and this can be done as quickly as in 1 to 2 hours, few hours, right? Now this is the basic technique that is being used for diagnosis of any disease. What do I mean by any disease? Means infectious disease. In COVID-19, we are using some modification of this called as RT-PCR because the genomic material for COVID-19 is RNA. And but not only for infectious particle or infectious agent, but why it becomes more important. For example, in early infection, when the copy number of the infectious agent is very low, copy number is low, then PCR can amplify and it can detect. So it has very high sensitivity. High sensitivity. So it can detect low amount of infection at a very early stage. Second, so this is infection. Second is that is that PCR can be used for cloning. So what is cloning? Cloning is just like, Xeroxing, where you generate identical copies of your notes or any literature. So Xeroxing, when you put your one single page on a Xerox machine, and you give instruction whether to print 5 or 10, and all the copies that comes out are just identical. So the spirit of cloning is to generate identical genes, right? And that is possible through PCR. The second is to look at the effect, right, of medicine. For example, let's say somebody had COVID, 1 week back, and now that person is fine and now wants to travel. So to make somebody safe for traveling or for an appointment, right? So one can do, again, go back to RT-PCR for getting positivity or negativity to ruling out. Second is effect of medicine. So if you're taking antiviral medication, medication to see whether it is working or not. So you will do the test PCR before and after. So before the medicine is administered and once the course is completed. So to look into that. Same thing can be applied for forensic. So we generally, if forensic scientists, they got a very small amount of DNA. And I mentioned to you that there are some genes which are same. Some genes are different in all of us, right? So the difference between us can help us in the situation of crime or any suspense or any clarity, right? Paternal suit or any dispute. Forensics can be very helpful if the amount of DNA or genetic material or tissue is very less and as low as hair follicle. So it can be used for forensic investigation also. So PCR is pretty powerful technology. And again, I want to go to the idea of Carey Mullis to tell you that PCR is actually nothing. Nothing in the means that it's a limited segment of DNA, because our chromosome, our DNA is meters long. It may be very difficult to replicate the whole set of 46 chromosomes, but it is definitely possible to do a patch of DNA to amplify. So by enzymes going back and forth here, it can help to generate millions of copies that we will see later. Now I mentioned that, back and forth. So what is the limitation? How do you define back and forth, right? So I use the word back and forth, right? So DNA is a parallel molecule, but not literally parallel. It is anti-parallel. So one strand is 5' to 3', and the other strand is 5' to 3'. DNA is anti-parallel because of the polarity of the sugar molecules, ribose molecule. Now, how to define? So the boundary is defined by the primers. So let's say, for sake of understanding, let's say this is how DNA is. Let's say there's a cell. There is a cell. This cell has a nucleus. In the nucleus, there is our DNA. So to get to the DNA, we have to first break the cell wall, get rid of all the unwanted protein, then destroy the nuclear membrane, and then get access to the DNA. and then this DNA is extracted. Now this DNA is like it's convoluted, but we are only interested in insulin gene. We are only interested in insulin gene. We don't care about other DNA. Why? Because our goal is to amplify insulin gene, and then we can do recombinant DNA technology to produce insulin. So the insulin gene is still here only. It's not all over the chromosome. So let's say this represents the insulin gene. So this is the extent of insulin gene. And we are only interested in amplifying insulin gene. So how to limit this boundary? So this is done by a pair of oligonucleotide, right? Oligonucleotide. And this is called as, these are pair of primers, right? So the boundary of a gene is defined in PCR by primers, the extent. We don't want, you have to choose your target. That has to be very, very specific. And then remember what did Carey Mullis think of? Back and forth, right? From this direction to this direction. Again, back and forth. And each time it happens, it generates one daughter DNA. Now, again, remember that the elongation, the DNA is synthesized in from 5' to 3'. Right? Now, the question comes. So one thing is that, one point I told you that the extent of a gene is defined by the primer. So and for example, if we are interested in amplifying 1 copy of insulin gene to million copies, then we have to select the extent of insulin gene to only magnify or amplify that, right? Now remember, in the beginning of the chapter, I mentioned to you that it's a blessing that the 2 strands of DNA, they are connected by hydrogen bond. They're connected by hydrogen bond, right? Because if they were connected by covalent bond, then it would have taken huge amount of energy to break this covalent bond. So because of this hydrogen bond, which is weak, if you take a DNA into the tube, take the DNA into a tube and raise the temperature to 95 degrees Celsius. So what will happen? This DNA will separate. How did this separate it? Because of hydrogen bond has broken disrupted under the high heat. Now what's the beauty? 95 degrees is a lot of temperature, very hot, right? So if you take proteins, lipids, carbohydrate, put it at 95 degrees, they all will turn brown, right? Very quickly, this is such hot. And even if you cool the temperature, they will not go back to the original form. But the beauty of the DNA is that when you raise the temperature, the DNA will separate, and when you lower the temperature, DNA will come back to its native structure. DNA will come back to its native structure. That's the beauty of DNA. So keep that in mind. So I use the word heat. So PCR is nothing but a thermostat. And what is thermostat? Thermostat, there is change in temperature. Change in temperature. Now, why there is change in temperature? Because a PCR operates in cycle. First at 95 degrees, second at 55 degrees, and third at 72 degrees. So what happens at this temperature? So at 95 degrees, DNA separates into single strand. I mentioned to you, hydrogen bond breaks. At 55 degree, primer anneals. And then at 72 degrees Celsius, new strand is synthesized. Now let's look this all the three steps as. So 95 degrees, when the DNA separates, it is called as denaturation. At 55 degrees, when the temperature comes down, the primer, the boundaries, the oligonucleotides attaches to the parent DNA. At 72 degrees, the new strand is synthesized. So let's look at as a let me explain to you with the pictures. So let's say this is the DNA. At 95 degrees, the DNA separates. And then temperature is reduced. Then this particular DNA tries to come closer to each other because it's no more degenerated. The temperature has been reduced from 92 to 55. But what happens that at this, as the temperature is lowering down, it also, the reaction, the PCR experiment also has primer. So these primers, they anneal to the boundary of the gene that has to be multiplied, to be amplified. So let me redraw this. So this is the situation, right? So here, and then this is the primer. Now you need something to synthesize the daughter DNA, right? You need something to synthesize the daughter DNA. What do you need? So number one, what do we need is ATP, CTP, GTP, GTP, and TTP. These are the building blocks, bricks of PCR, because oligonucleotide are important, right? To synthesize DNA, these are the building blocks. These are the bricks, just like our building has brick. So for DNA, this is the brick. Now, who will do the synthesis? So this synthesis is also done by a DNA polymerase. But this is a special DNA polymerase called as Taq DNA polymerase. Now, why special? Special because this DNA polymerase is derived from a bacteria called as Thermophilus aquaticus. Thermophilus aquaticus. Now, this bacteria lives in thermophilic environment. So, as the temperature is changing in PCR from 95 to 55 to 72, right? 95 denaturation, 55 annealing, 72 elongation. As the temperature is changing, right, it goes to 95 and then comes back to 55, primer annealing, then elongation, then again 95, then again 55, then again 72. So this goes in cycle. If we take human DNA polymerase, our DNA polymerase cannot stand this temperature. It will be charred, turn into charcoal right away. So this enzyme from a thermophilic bacteria helps to carry out the polymerase chain reaction. So let's go back to the slides. So this is essentially a PCR machine, right? This is old model, right? But now they're small, just like very small. You can keep it in your backpack, and they're much more stable. So this is a PCR machine. These are those pressing buttons where you can feed in the temperature, number of cycles. This is the tube holder, right? And this is the cap that you can shut once you have put the reaction, and just press the start button, forget, go back to your seat, have coffee, and it will come back. You come back and your gene is already amplified, right? So these are the uses of utilities or significance of PCR for cloning, for diagnostics, forensic treatment, and of course, genotyping. And so I'm just repeating what I draw. So we need these building blocks, GTP, CTP, TTP, ATP. And also we need enzyme. So what are the raw materials needed for? So the raw material is, of course, a target gene that we need to amplify. And the primers are the oligonucleotides that is complementary to the target gene to exactly amplify the gene you want, nowhere else. And then ultimately, Taq polymerase. Now, most of these enzymes, they need magnesium chloride as a cofactor for functioning. So you put all these things as a cocktail in a tube, PCR tube, target DNA, primers. How many primers? 2 primers. Taq DNA polymerase, the oligonucleotides, right? And you want to do it in an airtight space because sometimes there are many things in the air, especially in the lab. So PCR is very sensitive. So it's better to do it in an isolation airtight box. Then this is what I said, the magic of hydrogen bond. Three steps: denaturation, annealing, and some people say elongation, other people say extension when the new DNA is synthesized. So hydrogen bond is very, very important. Plays a very critical role in PCR. And this is the parent DNA. It separates the primers, bind P1, P2, the first cycle, the second cycle, and then it keeps on going for many cycles. For 1 hour, in 1 hour or 2 hours, it can generate millions of copies. The only thing that I feel I explained it as much as I could. I wanted to show a video, a very interesting video, but the only problem is that I can try, but sometimes it does not work. I don't know why, showing the YouTube videos. So I see that, and there are some questions in the chat area. So, okay. Yeah, so there is no, somebody asked me about this is chapter 5 or chapter 2. So it is technically chapter 5, but since I'm teaching on the second day, so sometimes I also mention it as a chapter 2. But the first chapter was history of DNA and all the basic information. And the second chapter itself or the second topic for the second week is chemical synthesis, PCR, and DNA sequencing. So I will finish. I had planned to finish PCR only for this class. Next class, your next class is on. So today is 15th. So we meet on 17th. On 17th, I will cover both DNA sequencing, gene synthesis, and mRNA platform, mRNA synthesis. Three topics. But if you understand PCR, the basics, the fundamentals, then the rest of the two or three topics will be very simple for you. And on 21st of Feb, you have your first quiz from the first chapter. Okay. And sorry, on 22nd Feb, I apologize. 22nd Feb, you have your first quiz, and you take the quiz. I'll give the whole time, but since this is synchronous, on 22nd February, there won't be any class, only quiz. Any questions?
Yes, Professor, could you please just show me what the book looks like, please, if you have it next to you?
Book? Okay.
Yeah, the text.
Just give me 1 second. So can you see here? I just showed you the book. Yes, Kaila Aditji, 1 second. Kaila Aditji, yes, the quiz will be available between 7 a.m. to 11:59 p.m. on 22nd February. Yes, Miss, you were asking some question about book?
No, I just want to see what it looks like. Thank you.
Okay. You are in my other class, right?
Correct.
Why don't you walk down to my office on Monday, then I can show you the physical copy.
Will do. Thank you.
Yeah, just remind me on that day.
Okay, no problem. Thanks.
Mm-hmm. Any other questions? And I try to always ask.
No, Professor. No other questions.
And I always try to ask, that if there are, if my teaching is not clear, then, feel free to send me an email, and you can suggest how do I can make it better. That's point number one. And second, those students who still have not done their introduction, they should go it. I have extended the day till the time till February 16th. You have, lot of time to do that. And if you don't do that, then by rule, I am supposed to mark you absent. So I don't want to do that. So make sure you are on top of it. And I've been reminding every week in every lecture. Any other questions?
No.
So if there are no questions, then if you wish, we can end the class.
All right. Thank you, Professor. Have a good night.
You too. Have a good night, all. Take care.
Thursday.
Yeah, Thursday.
Good night, Professor.
Good night. Take care.
Professor, I'm sorry, I didn't hear you. If you marked me.
Okay, I'll mark you. Okay, sure.
Thank you.
Welcome.